Good morning. We shall discuss today about the general structure and organization of bacteria. So, what is a bacteria? Bacteria is a single-celled microscopic organism that lack nuclei and other cell structures. So actually bacteria is a plural form. The singular form is called as bacterium. Bacterium is the singular form for bacteria and bacteria is the plural form. Generally we can say bacteria or pathogenic. Pathogenic meaning capable of causing disease. Several bacteria are pathogenic, that is, they are capable of causing disease, whereas most are non-infectious and they have many roles in fermentation, in nitrogen fixation, and in decay process and in nutrient recycling. These are the most bacteria which are non-infectious. They play a role in these various parts. So bacteria are generally called as gram positive bacteria or gram negative bacteria based on the basic microbial staining procedure. Based on the basic microbiological staining procedure, they can be called as either gram positive or gram negative. The test that is used is called as gram staining test. And bacteria generally come in several shapes and sizes. So if we see the generalized structure of a bacteria, the bacteria looks like this. So it has this outer one is called capsule, the inner one is called cell wall, and these are called phloem, and this is a chromosomal DNA, and this is called pilus. These are called plasmid DNA. These are called ribosomes, and this is called cytoplasmic inclusion. So this is the generalized structure of a bacterium, that is a singular form of bacteria, and there are various shapes of bacteria. one shape is like this. These are called as micrococonuts. And there is one other shape like this. These shapes are called as Diplococcus. And we have one other shape where it looks like a cluster like this, and these are called as Staphylococcus. Then we have it like a strand like this, and these strands are called as Streptococcus. So these are few shapes of bacteria. There are few more shapes which are called as Corynebacterium, Bacillus typhi, Protus, Spirillum, Syphilibacteria, nitrogen feeding bacteria, Thysophilium, and Vibrio. Those are different types of different shapes of bacteria that we can see. There are different sizes of bacteria. Bacteria generally has a diameter which ranges from 0.75 to 1.25 micrometer and an average of 1 micrometer. Whereas if you take Bacilli type has varying length from 2 to 10 times their width. If we take Bacilli, they are very short Bacillus type. Then the next one is filaments. These are long threads of bacilli which have not separated into single cell. Okay, so we have seen here what is meant by the singular form, what is the bacteria, then how are bacteria classified, they are pathogenic or whether they are non-infectious. Then bacteria are classified as gram positive, gram negative based upon the staining procedure that is used. Then the different shapes and size. This is the general structure of how a bacterium looks like. And then we had seen different shapes. So the first one is the Micrococcus, Diplococcus, Staphylococcus, and Streptococcus. Then based on size, there are furthermore classification based upon these shapes. Then based on size, we have classified it into 4 different types. Now we'll see where and all bacteria is found. Bacteria is generally found everywhere under the earth to a distance of 400 meter in the air at high altitude and at high temperature. So we can see that bacteria are generally found in almost everywhere, and it can go under the earth up to 400 meter, and it can be seen at high altitude in the air, and it can also be seen in high temperatures also. And animals, if you see in the animals digestive system and in the animals respiratory systems also, these bacteria are there so that it will be able to produce some digestion process. So we had seen here the various parts that is there in a bacteria. First we will start with the flagellum. Flagellum, we call it as flagella. The singular form is called as flagellum, and these flagellas are made up of class of linear proteins which are called as flagellines. The basal body traverses the cell wall and cytoplasmic membrane to anchor the flagellum, and it will enable it to veer about its axis like a propeller. The function is, this flagella's function is to give the bacteria the ability to move about the activity. Then next is the capsule. We had seen in this figure, this part is called as capsule. So these capsules are generally consisting of, most bacteria will be consisting the capsule as a polysaccharide, and the function of this is it will get attachment to surface, and it will be acting as a protection against engulfment, and it reserves of nutrients. These are the various functions of the capsule. Next is the plasma membrane that we have seen in the structure. The plasma membrane will be composed of phospholipids, which will be containing polar group attached to 3 carbon glycerol backbone. So this is a plasma membrane. Again, this plasma membrane we have seen in the figure over here. So this plasma membrane will be having this kind of composed of phospholipids, which will be containing polar group attached to 3 carbon glycerol backbone. And the function of this plasma membrane is for selectively permeable, in that it helps to control what moves into and out of its cell. So this is the function of a plasma membrane. Then there is a plasmid. Plasmid is a cellular nucleus. Okay, this plasmid is a cellular nucleus which consists of a tangle of double strand DNA. What we are seeing here, we are seeing here a DNA, right? This DNA is that. So this particularly has a long DNA or protein fiber which will be called as chromatin. Each fiber will build a so-called chromosome. The plasmid of human pathogen bacteria often will bear important genes determining the phenotype of their cell, that is the resistant genes or virulence genes, etc. Then there are cytoplasm and ribosomes that are there in the bacterium, and the function of ribosomes are, as I told, other than these we have ribosomes and cytoplasm that is seen in the structure of a bacteria. Ribosomes are acting as organelles for protein synthesis, whereas cytoplasm are going to be consisting of large number of solute, low and high molecular weight substance. So The cytoplasm has the RNA and approximately 20,000 ribosomes or cells. Bacteria usually have 70 as ribosomes, which comprises totally it will be having 70 ribosomes, which will be comprising of 30s and 50s subunits. Then next is a cell wall. The cell wall of a bacterium. Okay, the most important structural element of the cell wall is called as murine. Murine is a net-like polymer material which surrounds the entire succulus. This murine is going to be made up of polysaccharide chains cross-linked by peptides. Okay, so the cell wall is going to make use of most important structural element of the wall is the murine. Murine is a net-like polymer material which surrounds the entire succulus, and polysaccharide chains cross-linked by the pesticides. And basically we are going to have cell walls. The cell walls function is to maintain the overall shape of bacterial cell, and the cell wall will protect the cell protoplast from any mechanical damage, and it will also protect it from osmotic rupture or lysis. So these are the functions of a cell wall of a bacteria. And this is the basic structure of a bacteria what we have seen. So we had started with what is meant by bacterium and what is meant by bacteria, and after seeing this too we had just discussed about what is meant by pathogenic and non-infectious and how bacteria are classified based upon the staining procedure, then based upon the shapes, based upon the size. Then we had seen the basic structure of a bacterium and the various parts that are there in the structure of bacterium and what are the various functions. So that is what we have discussed today in detail. Thank you for listening.